After reading and understanding this module, the technique of mass spectrometry can be used to determine the molecular mass of proteins. Proteins which are fragmented by proteases can be separated by HPLC or two-dimensional electrophoresis and then analyzed by mass spectrometry and thereby the sequence deduced. Mass spectrometry is a powerful and fast technique in the analysis of proteins and peptides, although at present very large proteins cannot be analyzed by this technique. Let me introduce you to this major technique. The technique of mass spectrometry developed due to the Nobel Prize winning discovery of Sir J.J. Thomson, who in 1897 discovered the electron and observed that ions in the cathode ray tubes moved with trajectories proportional to their mass to charge ratios or MZ ratios. Although initially mass spectrometry was only applicable to smaller volatile ions, in the 1950s, the 1980s, new developments in the techniques of volatilizing small proteins and peptides were developed, allowing these to be thereby analyzed by mass spectrometry. Today, the technique can be used in time frames of minutes to sequence small peptides, unlike traditional methods prevailing like the Edman sequencing technique, which takes more than an hour for an average size peptide. Furthermore, molecular masses of these peptides, proteins can be determined with an accuracy of 0.01%. Let us go through the technique of mass spectrometry. A mass spectrometer analyzes volatile ions which are generated by ionization of the analytes in vacuum and then passing them through a mass analyzer having an electric and or magnetic field, a cathode ray tube, wherein they separate based on their MZ ratio. The MZ spectrum is then deconvoluted to determine the exact mass of the ion. The principle of a mass spectrometer is best explained by studying the figure. A stream of the volatilized sample is bombarded by an electron beam which generates positive ions. The ions are accelerated and passed through a magnetic field and filter which removes the larger and smaller ions into a mass analyzer which gives a spectrum as indicated in the figure. Based on relative abundance, the spectrum is deconvoluted to read the molecular mass within the range of plus or minus of the real mass. Now, let us try to understand the techniques applied to volatilize proteins and peptides. Till the 80s, mass spectrometric analysis of hydrophilic molecules such as peptides and nucleic acids were not possible because these could not be volatilized into the gaseous phase, which was a necessary prerequisite for mass spectrometry. Two major techniques were developed, ESI and MALD, which were successful in volatilizing proteins and peptides. Let us look at the first mentioned technique of volatilization, namely electrospray ionization, or better known as ESI. This process, developed by John Fenn, involves injecting a macromolecular solution through a capillary tube under high pressure and maintained at a voltage upwards of 4000 volts so that the sample comes out as a spray. The spray contains fine droplets of the macromolecules from which the solvent rapidly evaporates as a stream of dry nitrogen or any other gas is passed. As the solvent evaporates, the peptide or analyte is in a gaseous ionic form with a charge of plus 0.5 to plus 2 per kilodalton, and these can then be analyzed by the mass spectrometer. The principle of ESI is best illustrated in the alongside diagram. Now let us look at the other technique used to volatilize proteins, matrix assisted laser desorption ionization or MALD. In this procedure, introduced in 1988 by Hillenkamp and Karras, the macromolecule or peptide is embedded in a crystalline matrix of a low organic mass such as gentisic acid, which is 2,5-hydroxybenzoic acid. The crystalline material is then irradiated with a short note nanosecond pulse of a laser light of wavelength absorbed by gentisic acid. The high energy attained by the gentisic acid or matrix causes the peptide to be ejected into the gaseous phase with a charge varying from plus 1 to plus 3. These ions are then analyzed by the mass spectrometer. Peptides and polypeptides of mass around 400 kilodaltons have been analyzed by MALD. The matrix such as gentisic acid is a UV absorbing weak organic acid which also acts as a proton donor to the peptide. How do we analyze a mass spectrum? In the indicated spectrum, the rightmost peak would correspond to the ion with the lowest charge, therefore highest MZ ratio. If the charge or Z value of the ion is known, its mass can be more accurately determined. Alternatively, if the MZ values of two adjacent peaks of a peptide of mass M are alpha 1 and alpha 2 arising from ions of Z1 and Z1 minus 1, then two linear equations can be derived from which the value of M and Z1 can be derived. Note the two equations are as follows: alpha 1 is equal to M plus Z1 by Z1, equation 1. Alpha 2 is equal to M plus brackets Z1 minus 1 divided by Z1 minus 1, which is equation 2. Both these are linear equations which can be solved for M. Now let us look at the applications of mass spectrometry. The sensitivity and speed of mass spectrometry has revolutionized analysis of molecules. With the development of ESI and MALD, due to which proteins and nucleic acids can be volatilized, the applications in biochemistry are immense and evolving. In this section, a few important applications in the analysis of proteins are illustrated. Let us look at tandem mass spectrometry in sequencing peptides. Initially, a larger peptide is fragmented with an enzyme or randomly to generate random overlap fragments. Two mass spectrometers are placed in tandem, the first selecting a given peptide ion from other ions and passing it to a collision cell where the peptide is bombarded by an inert gas to generate a few fragments, and these are then analyzed for masses by the second mass spectrometer. The first mass spectrometer separates the peptides and the second analyzes the selected peptide which is fragmented in the collision cell. each peptide of the selected fragment from the first mass spectrometer is broken in only one place, usually at the peptide bond. The second mass spectrometer generates one or more sets of peaks, one set of peaks wherein the charge was retained in the N-terminal fragment, which is also known as the B-type fragments, and the other set corresponding to the charge retained on the C-terminal fragments known as the T-type fragments. If the masses of all the peptide fragments are determined, a comparison of the molecular masses of successively larger fragments can easily be fitted with known molecular masses of amino acids, except in the cases of isomers such as isoleucine and leucine and glutamine and lysine. The latter set differ by only 0.036 Daltons. This comparison process has been programmed to compute sequences within minutes of the mass spectrometric data. Furthermore, a database of known mass spectrum of several peptides are available, from which new obtained data can be matched and confirmed. It has been demonstrated that the determination of a mythical peptide sequence from MS-MS data. The selected peptide from MS1, when passed through a collision cell, is cleaved and generates N-terminal peptides and C-terminal peptides as indicated. A random sequence assigned AD to VR is broken to give an N-term B-type kind of peptide, N-terminal, and a Y-type or T-type peptide, which is the C-terminal peptide. Observe the figure which gives a spectrum of the analyzed peptides from the mythical polypeptide. Plotted on the y-axis is the relative intensity and on the x-axis the MZ ratio. I hope it is obvious that more right in the graph refers to basically the smaller charge and therefore the higher MZ ratio. One of the advantages of MS-MS sequencing is that peptides with blocked N-termini or other post-translational changes which are not possible to sequence by the Edman procedure can yet be analyzed. The Edman procedure is a chemical process which requires a free N-terminus. So, this advantage is there for the MS-MS kind of analysis because it does not require the presence of a free N-terminus. Furthermore, the quantities of peptides required for MS-MS fall in the nanogram to picogram range, which makes the technique rapid as well as sensitive. Let us now analyze another kind of application which the MS-MS technique can be used for, namely analysis of 2D gels in proteomics. In proteomics research, the entire proteome, namely all proteins made of a cell, is examined under different conditions such as heat stress or any other diseased condition. This analysis would provide clues to what factors are involved in creating that condition. Two-dimensional gel electrophoresis is a sensitive and powerful technique to analyze the proteome. It is known as two-dimensional because in the first dimension, proteins or peptides are separated based on their isoelectric point or pH. In the second dimension, they are separated based on their molecular weight. In the first step of two-dimensional electrophoresis, proteins and polypeptides are extracted from cells under a given set of conditions and then are separated from one another by their isoelectric pH in capillary tubes. This is possible by using low molecular weight organic acids and bases, namely ampholytes, in the capillary tube containing the polyacrylamide gel. The ampholytes distributed in an applied electric field is indicated in the figure, and these set up a pH gradient. The separating polypeptides come to rest wherein their respective isoelectric pH matches that of the ampholyte. In the second step, the capillary gel is placed over a second SDS-Page slab gel, and the polypeptides move in the second dimension based on their molecular weight. It is possible using robotic arms to pick up each protein spot after two-dimensional electrophoresis and submit them to mass spectrometry. Hence, the proteome can be analyzed by the molecular weights of the various proteins. Databases of various proteomes can be maintained and available for comparison. The figure illustrates the use of mass spectrometry in analyzing a proteome. In the figure which has been illustrated, this indicates the MS analysis of a proteome after 2D analysis from a normal and diabetic mouse. Differential protein spots obtained after comparing 2D pattern of proteomes, that is the proteome from a normal mouse and that from a diabetic mouse, can be analyzed by MS for not only molecular masses but also by MS for sequence information, and comparison with databases can even identify the nature of the protein which has been expressed differentially in the normal and the diabetic mouse. Now let us look at some of the future applications of mass spectrometry. Mass spectrometry has now been extended to the analysis of nucleic acids, virus particles, protein nucleic acid complexes and other non-covalent biological complexes, as long as the polypeptide complex can be selected from other contaminants, its mass and sequence can be determined. Note the figure in which the blood from a newborn can be screened for containing sickle cell hemoglobin or any other molecular variant of the normal hemoglobin within minutes just using a drop of blood. This could be important information for the neonatologist in the course of treatment to be followed. This of course can be extended to the determination of the molecular masses of proteins or other analytes in inherited disorder diagnosis. Let me summarize what you have learnt in this module. First of all, mass spectrometry has become a rapid and sensitive tool to analyze proteins and polypeptides. The development of ESI and MALD for the generation of gas phase ions of peptides has been instrumental in the development of the technique in proteomics applications. Novel applications of MS, such as that referred to earlier in the analysis of neonatal blood, are yet to reach their potential, such as protein nucleic acid associations and also neonatal diagnosis.